Chapter 2, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using the criteria that were scientific, but born out of need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those had red blood and those that did not. Linnaeus in Linnaeus' time, a two-kingdom classification system with Planta and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between eukaryotic and prokaryotic, unicellular and multicellular organisms, photosynthetic green algae, and non-photosynthetic fungi organisms. Classification of organisms into plant and animals were easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for long period of time was found inadequate. Beside gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, and evolutionary relationships, etc. Classification system for living organisms have hence undergone several changes over the time. Though plants and animal kingdoms have been a constant under different systems, the understanding of what groups or organisms to be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been understood differently by different scientists over the time. R. H. Whittaker, 1969, proposed five kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a six kingdom classification. You will learn about this system in detail in higher classes. Let's look at the five kingdom classification to understand the issues and considerations that influence the classification system. Earlier, the classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. The character that united this whole kingdom was that all organisms included had cell wall in their cells. This placed together groups which widely differed in their other characteristics. It brought together prokaryotic bacteria and blue-green algae cyanobacteria with other groups that were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogara were placed together under algae. The classification did not differentiate between heterotrophic group, fungi, and autotrophic group, green plants, though they showed a characteristic difference in their wall composition. Fungi had chitin in their walls, while green plants had cellulose in their wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista had brought together Chlamydomonas, chlorella, earlier placed in algae with plant, and both having cell walls, with Paramecium and amoeba, which were earlier placed in the kingdom Animalia, which had lack cell wall. It has put together organisms which in earlier considerations, earlier classifications were placed in different kingdoms. This happened because criteria of classification have changed. This kind of changes will take place in future too, depending on the improvements in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only on the morphological, physiological, and reproductive similarities, but also in phylogenetic characters, which is based on evolutionary relationships. In this chapter, we will study characteristic of kingdom Monera, Protista, fungi, or Whittaker system of classification. The kingdoms Planta and Animalia, commonly refers to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole members of kingdom Monera. They are most abundant organisms. Bacteria occur almost everywhere. Thousands of bacteria, hundreds of bacteria are placed in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on organisms as parasites. Bacteria are grouped under four categories based on their shape: spiral coccus, rod shaped bacillus, comma shaped vibrio, and spiral shaped spirillum. Though bacteria structure is very simple, they are very complex in their behavior. As compared to many other organisms, bacteria as group show most extensive metabolic diversity. Some of the bacteria are autotroph. They synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotroph. They depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in guts of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have a chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example: Nostoc and Amina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrites, nitrates, ammonia, and they use released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority the majority are important decomposers. Many of them have significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legumes, roots, etc. Some are pathogen causing damage to human beings, crops, farms, animals, pets. Chlorella typhi, tetanus, citrus canker are well-known disease caused by these bacteria, different bacteria. Bacteria reproduce mainly by fission, sometimes under unfavorable conditions they produce spores. They also reproduce by short of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another bacterium. The mycoplasma are the organisms that completely lack a cell wall. They are smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic to both plants and animals. Kingdom Protista. All single cell eukaryotes are placed in the Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to other. In this book, we must include chrysophytes, dinoflagellates, euglenoids, slime mold, and protozoan under Protista. Member of Protista are primarily aquatic. This kingdom form link with others dealing with plants, animals, and fungi. Being eukaryotic, Protestant cell body contain a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually or sexually by the process of asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. This group includes diatoms and golden algae dismids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic. In diatom, the cell wall form two thin overlapping cells which fit together in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers of oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, red, and depending on the main pigment present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them has two flagella. One lies longitudinally and other transversely in a furrow between the wall plates. Very often, red dinoflagella example Goniolax undergo such rapid multiplication that make the seed appear red. Red tides, toxin released by such a large number may kill other marine animals as well, such as fishes. Euglenoids. Majority of them are freshwater animals found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, short and long one. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on small animals. Interestingly, the pigments of euglenoids are identical to those present in higher plants, example Euglena. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. Under unfavorable conditions, the plasmodium differentiate and form fruiting bodies bearing spores at their tips. The spores possess true walls, they are extremely resistant and survive for many years under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs. They live as predators or parasites. They are believed to be primitive relatives of animals. There are four main major types of protozoans. Amoeboid protozoans. These organisms live in fresh water mostly, seawater or moist soils. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. The marine forms have silica on their surface, and some of them, such as ant amoeba, are parasites. Flagellated protozoan. The members of this group are either free living or parasitic. They have flagella, and the parasitic form causes diseases such as sleeping sickness. Trypanosoma. The ciliated protozoans, these are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with food to be steered into the gullet. For example, paramecium. Sporozoans. This include diverse organisms that have infectious spore-like stage in their life cycle, and the notorious animal in this group is Plasmodium, the malarial parasite which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. Now, the kingdom fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on moist bread and rotten fruits. The most common mushroom that you eat and the toadstool are also fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example yeast, are also used to make bread and beer. Other fungi cause disease to plants and animals. Wheat rusting puccinia is also an example. Some of them are source of antibiotics like penicillin. Fungi cosmopolitan and occur in air, water, soil, on animals and plants as well. They prefer to grow in warm, humid places. Have you ever wondered why you keep food in refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infection. With an exception of yeast, which are unicellular, fungi are always multicellular. Mostly, their bodies consist of long, slender, thread-like structures called hyphae. A network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called sinusotic hyphae. Others have septate or cross wall in their hyphae, and the cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic, absorb soluble organic matter from dead substrates and are called saprophytes. Those that depend on living organisms and animals, plants, they are called parasites. They can also live as symbiotes in the association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means like in fragmentation, fusion, and budding. The asexual reproduction is by spores called conidia, sporangiospore, and zoospore. Sexual reproduction is by oospores, ascospore, and basidiospore. Various spores are produced in distinct structure called fruiting body, and sexual cycle involves three steps: fusion of protoplasm between non-motile or motile gametes called plasmogamy, and the fusion of two nuclei called karyogamy, and the last one, which is meiosis in zygote, resulting in haploid spores. When fungus reproduce sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, fusion of two haploid cells immediately results in diploid cell. However, in other fungus like Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus N 2 nuclei per cell occurs. Such dikaryotic stage condition is called dikaryon and the phase is called dikaryophase of fungus. The later parental nuclei fuse and the cell become diploid. The fungus form fruiting bodies in which reductional division occur and leading to formation of haploid spores. The morphology of mycelium, mode of spore formation and fruiting body form the basis of division of kingdom into various classes like Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Let's start with Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places and as obligate parasites on plants. The mycelium is aseptate and sinusotic. Asexual reproduction takes place by zoospores, which are motile, or by aplanospore, non-motile. These spores are endogenously produced in sporangium. And the zygospore is formed by fusion of two gametes, and these gametes are similar in morphology, isogamous, or dissimilar, anisogamous or ogamous. Some common examples are Michael, Rhizopus, Albugo, parasitic fungus on mustard. Ascomycetes, commonly known as sac fungi, Ascomycetes are mostly multicellular penicillium or rarely unicellular like yeast. They are saprophytic decomposers, parasitic or coprophilous. Mycelium of Ascomycetes is branched and septate. The asexual spores are conidia produced exogenously on special mycelium called conidiophore. The conidia germination produce mycelium and sexual spore called ascospore, which are endogenously produced in a sac like asci. These asci are arranged in different types of fruiting bodies called ascocarp. Some examples are aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. Bascomycetes, commonly known form of Bascomycetes are mushrooms, bracket fungi, or puffballs. They grow in soil, on logs, or tree stumps, and in living plant bodies as parasite as well. like rusts and smuts. The mycelium is branched and separate. Asexual spores are generally not found, but vegetative reproduction is by fragmentation that is common. Sex organs are absent, but plasmogamy is brought together by fusion of two vegetative or somatic cells of different stains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium, and the karyogamy and meiosis takes place in basidium, producing four basidiospores. And the basidiospore are exogenously produced on the basidium. And the basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus, mushroom, ustilago, smut, and Puccinia rust fungus. Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and sexual stage given another name and placed under another class. Later, when the linkages were established, the fungus were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of the members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce asexual spores, only by asexual spores, known as conidia, and the mycelium is separate and branched. Some members are saprophytes or parasitic, while the large number of them are decomposers of litter and help in mineral cycling. Examples are Alternaria, Coleotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll containing organisms commonly called plants. Few members are partially heterotrophic, such as insectivorous plants or parasites. The bladder volt, the Venus flytraps are the examples of insectivorous plants, and the cuscuta is the parasite. The plant cell have eukaryotic stage with prominence of chloroplast and cell wall made up of cellulose. You will study that eukaryotic cell structure in detail in chapter 8. The plant include algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plant has two distinct phases: the diploid sporophytic phase and haploid gametophytic phase that alternate with each other. The length of the sporophyte, haploid and diploid phases, and whether these phases are free or dependent on other, vary among different groups in plants. This phenomena is called alternation of generation. You will study about these further in detail in chapter 3. Animal kingdom. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell wall lack cell wall. They directly or indirectly depend on plants for food. They digest their food in internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have different shape and size. High forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. Structural sexual reproduction is by copulation of male and female, followed by embryological development. Salient features of various phyla are described in chapter 4. Viruses, viroids, and prions. In 5 kingdom classification of Whittaker, there is no mention of lichens and acellular organisms like virus, viroids, and prions. They are briefly introduced here. All of us who have suffered from the ill effects of common flu know what other effects viruses can have on us, even if we do not associate it with our condition. Viruses do not find any place in classification since they are not considered as truly living. If we understand living as those that have cell structure, viruses are not cellular organisms that are characterized by having an inert crystalline structure outside their living wall, living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non living? See, viruses mean venom or poisonous fluid. Dmitry Ivanovsky, 1892, recognized a certain microbes as causal organism of mosaic disease tobacco. They were found to be very small than bacteria because they are passed through Bacteria-proof filters. M.W. Bijernik, 1898, demonstrated that extract of infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid Contagium vivum fluidum, the infectious living fluid. W. M. Stanley, 1935, showed that viruses could be crystallized and the crystals contain largely proteins. They are inert outside their specific host cell and the virus is an obligate parasite. In addition to protein, viruses can also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. Virus is a nucleic protein, nucleoprotein, and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and the viruses that infect animals have either single-strand RNA, double-strand RNA, or double-strand DNA. Bacterial virus, bacteriophages, bacteria that infect viruses that infect bacteria are usually having double-strand DNA virus. The protein coat called capsid made up of small subunits called capsomer protect the nucleic acid and these capsomers are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes and influenza. AIDS in humans are also caused by virus and in plants the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunted growth. Viroids in 1971, Theo Denier found and discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. PSTD. It was found to be a free RNA and it lacked protein code that was found in viruses, hence called viroid. The RNA of viroid was of low molecular weight. Prion. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to virus, and these agents were called prions. The most notable disease caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant C. Jacob disease, CJD, in humans. Lichens are symbiotic association, mutually useful association between algae and fungus. The algal component is known as phycobiont and the fungal component is called mycobiont, which are autotrophic and heterotrophic respectively. Algae provide prepare food for fungi and fungi provide shelter and absorb minerals, nutrients, water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within it. Lichen are very good pollution indicator that do not grow in polluted area. Summary. Biological classification of plants and animals were first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, plants and animals. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, fungi, plants and animals. The main criteria of the five kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In five kingdom classification, bacteria were included in kingdom Monera. Bacteria are cosmopolitan in distribution, and these organisms show most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-cell eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have definite, well-defined nucleus and other member organelles. They reproduce both asexually and sexually. Member of kingdom fungi show great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes are four classes under this kingdom. The plant kingdom include all eukaryotic chlorophyll containing organisms. Algae, bryophytes, pseudophytes, gymnosperms and angiosperms are included in this group. The life cycle of plant exhibit alternation of generation, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organism lacking a cell wall are included in the kingdom Animalia. Thank you guys for watching this video. Bye.